Thank you. Wonderful panel and wonderful speakers today. In comparing results from your rotator cuff versus liposuction studies, have you taken into account the damage of the adipose cells during the liposuction removal process?
Great first question. And the answer is no. It's really difficult to get these same sort of samples where, say, in the rotator cuff, those fat samples aren't necessarily damaged from younger patients. I mean, people with sports injuries certainly tear them, but we haven't yet gotten a sufficient number of younger patients from those types of surgeries to be able to make those sorts of comparisons. So there certainly could be a damage aspect to why we see younger patients that are undergoing liposuction not doing necessarily as well as our older patients. But it could also be then those anatomical differences as well. And the fat differences. They're completely different types of fat.
Thank you. And just one other short question. Do you favor any particular method of harvest, or are you looking at any new technologies? Or
we're really sort of beholden to our surgical colleagues. And I would say that they're sort of at the cutting edge of taking all these biopsies. I would say that we don't just biopsy the fat, but we get skeletal muscle samples as well, and we're comparing intraspinatus and supraspinatus muscles versus the deltoids, and it all just sort of depends on the tear state. So we haven't yet sort of pushed the envelope beyond what is sort of this clinical standard right now with that.
There was another one over there, Laura.
I have a quick question for Professor Goldstein. I noticed on your graph, looking at SERL1 suppression of A-beta peptide accumulation, it's 1 to 40. So does it work for the 1 to 42, and does that work for the Swedish mutation?
Okay, so that's an excellent question. For those of you who are not into peptide biology, there are some forms of the peptide that are just a touch longer that are thought to be more toxic, which is the 1 to 42 class. We don't have measurements of 1 to 42 on all of the samples because we're working with very small numbers of cells and high sensitivity assays. But in the handful we've been able to study and that others have studied, yes, it increases both. They're probably going to the same processing compartment, we think. And then the second question was, are there analogous effects on Swedish mutations? We don't know the answer to that yet. We're busy building cells where we've knocked Swedish mutations into, say, the Venter background, and then we'll see how it responds. I suspect it will, but we don't know yet for sure. Can I ask a question? Yeah, I want to ask a question of Adam. It's all right. So Adam, so when you take these mesenchymal progenitors and you expose them to surfaces of different stiffness, are those irreversible effects on their differentiation capacity, or do they have to be maintained continuously under stiff conditions to move down that pathway?
Very good question. And from the transdifferentiation assays that we have, we know that there's sort of a certain amount of memory that these cells have, or sort of ability to forget where they've been.
Well, a commitment, right?
Yeah, so commitment, exactly. Yeah. So if you were to do these sorts of lineage switching assays after about a week in culture, those cells are very plastic, unless they've fused together, and they'll transdifferentiate. If you wait longer and longer, then they sort of become less and less plastic. So there are matrices that people have made where you can change the stiffness as a function of time. So you can make an environment soft, and then with a quick dose of UV, you can cross-link everything and make it stiff. So those same cells went from thinking that they were soft and becoming fat to then it becoming really stiff. And depending on when you make that transition, you can sort of trick the cells to either transdifferentiate or not. Yes, no.
There's one up here, actually. Yeah. An ethical problem?
Yes, I actually have a question about for the brain work that a number of you, including Anne, who is, I think, not here anymore. She's here too. Well, this may be as much a question for her. I'm interested in the delivery system for talking about these various strategies with dealing with brain disorders like Alzheimer's, because a lot of the work you're doing is obviously on a mouse. You can't do the same thing with a human brain. So I'm wondering down the road how this is going to be handled from both a scientific and an ethical perspective?
That's definitely a question for Anne.
Do you want to take, well, just give that to me. I actually just had a conversation with Anne about that. But
So, you know, we have a neurosurgeon on staff, and from a regulatory perspective, if you deliver the cells with a standard neurosurgical instrument, it's not, you know, a new product or a device. So what you need to show is that your cells are compatible with that particular device, whether it be a catheter or a needle or syringe. So what we've tried to do, because we don't think we need a device to deliver the cells into the brain or the spinal cord or eye, we've worked with the people who perform surgeries and use instrumentations that they feel where they would have the best control over. So.
the Society for Neuroscience, of course, meets here in November. The day before that, on the 7th, the International Society for Neuroethics is meeting, and so those are some of the issues that will come up. In addition to having done the stem cell meeting on the Mesa, we're also talking about doing the brain meeting, and that's one of the issues that would have come up. In the early days of the stem cell meeting on the Mesa, as Larry will remember, we actually had ethics panels to talk about all of that, so I'm hoping that the President's Commission on Bioethics, Amy Gutman, and maybe Nita Farahani will be able to come here and talk about those sorts of issues as well. So.
Well, I think we're actually out of time.
Yeah, I'm just thinking about when I was saying it when the introductory remarks about the tenderness. I mean, the way in which you've all been saying, "Oh, that's an interesting question. That's an excellent question." It's really very, very courteous. The beyond belief meetings that we used to do, people like Lawrence Krauss would say, "Well, that's a really stupid question." So, but yes, great.